Honorable laureates, excellencies, distinguished colleagues, ladies and gentlemen, welcome to the Nobel Lectures in Chemistry 2024. So this year's Nobel Prize in Chemistry recognizes groundbreaking achievements in two different areas: computational protein design and protein structure prediction. So it's all about protein structures. As you may know, the polypeptide chain of a protein folds itself up in space into a complex three-dimensional structure. Now, the remarkable thing is that this 3D structure is somehow encoded in the sequence of amino acids that make up the chain. This finding that a linear sequence of amino acids dictates the 3D structure, it dates back to the work of Christian Amundsen, who received the Nobel Prize in Chemistry in 1972, so over 50 years ago. It meant that it should in principle be possible to predict the 3D structure directly from the sequence. Likewise, it should be possible to predict which sequences would produce a certain 3D structure. So it has long been a dream of biochemists to computationally solve these two problems. And it is clear that one needs to use computers to do it. The latter challenge, to predict sequence from structure, that's what protein design is all about. The idea is to draw a new protein, typically one that has never been seen before in nature, and somehow compute a sequence that will give this structure. The sequence could then be made in the lab, and completely new proteins with desired functions could thus be created. So this challenge was solved by David Baker and co-workers in 2003. They designed an entirely new protein with 93 amino acids and computationally predicted a sequence that would give this structure. They then made the protein and verified experimentally that the prediction was correct. The reverse problem, to predict 3D structure from sequence, is an astronomically huge search problem. And this was pointed out by Cyrus Levinthal already in the late 1960s. So progress in this field was slow indeed for many years. But Demis, Hassabis, and John Jumper and their co-workers, they solved it in year 2020 by ingenious neural network engineering, which resulted in the now famous program AlphaFold 2, and it was published in 2021. This was undoubtedly the real breakthrough in protein structure prediction, and AlphaFold 2 gave unprecedented accuracy. So this network is trained using the data stored in sequence and structure databases to discover correlations and patterns among amino acid sequences. And this allows it to produce accurate distance maps that can then be converted into 3D structures. We'll probably hear more about it from the laureates. So the work of this year's laureates has had an enormous impact. David Baker has opened up completely new possibilities to design proteins that have never been seen before, with many exciting applications in pharmacy, medicine, nanoscience, etc. And likewise, the work of Demis Hassabis and John Jumper has caused a revolution in structural biochemistry, and we now have access to predicted structures of all 200 million known proteins. And this is a phenomenal resource for biochemical and biological research, also with many new applications. So our first speaker, David Baker, he was born 1962 in Seattle, Washington, United States. He got his PhD in 1989 from University of California at Berkeley in California. and he is presently a professor at University of Washington, Seattle, and an investigator of Howard Hughes Medical Institutes. So please join me now in welcoming Professor David Baker for his Nobel lecture.
All right. Well, first of all, thanks everyone for being here. It's really wonderful. It's a great honor, and it's wonderful to see so many friends and family and colleagues. So today I'm going to tell you about de novo protein design. This is an artist's depiction of the interior of a cell in our bodies, and each of these colorful blobs is a protein, and each protein is carrying out a unique job or function. And indeed, in all living things, proteins are carrying out, they're basically the miniature machines that are carrying out all the important functions. And they evolved over hundreds of millions of years or billions of years to be really perfect at solving the problems that arose during biological evolution. But today, we face new problems. We live longer, so neurodegenerative disease and other problems now are arising. We're heating up and polluting the planet. And so if there was selective pressure over a really long time, new proteins might evolve to solve these problems, but it would take a very, very long time and not be very pleasant. The question the goal of de novo protein design is to make new proteins that can solve modern day problems, as well as the proteins that came through evolution solve the problems that were relevant during evolution. So first I need to remind you of a little bit of biology, and you already heard this, but I'll just give you the basics again. The genes in our genomes, each gene encodes one protein. It does so by specifying the amino acid sequence of the protein, which then folds up to the protein with which has a particular structure and biological function. So in de novo protein design, the flow of information is just the opposite. We start with a desired function that doesn't exist. We build a protein which we predict will have this function, and then compute the amino acid sequence this protein should have. And then this is a brand new protein, so there's no gene in nature which already encodes it. Instead, we make a synthetic gene, a synthetic piece of DNA, and that encodes this protein. We put it into bacteria, and the bacteria basically become factories for making the protein. We can get the protein out and then test to see whether it actually does what we designed it to do. And so today, I'm going to be giving you a number of different examples of designed proteins that we've designed to have different functions. And in every case, we've gone through this computation and then made a synthetic gene and then made the protein, and I'll be showing you what those proteins do. Let's see. So there is a huge space of possible proteins. And so there are 20 different amino acids, and a typical protein is a sequence of about 100 or more amino acids. And so that means the number of possibilities is 20 times 20 times 20, 100 times, or 300 times, depending on how long the protein is. And this is an astronomical number. And it's kind of depicted by this gray space here. Now, what's been sampled during evolution is a tiny, tiny fraction of the possibilities. And the proteins in nature are kind of indicated by these red ovals. These proteins, evolution proceeds incrementally. So proteins tend to be very closely related. The proteins in us are closely related to the proteins in other mammals and so forth. So it kind of bunches up a lot. So traditionally, and this is really up until the present, if an engineer or a biologist wanted to solve a new problem and they thought a new protein could work, the way it was done, and is still being done, is to look all through nature to see if a protein can be found that has that activity. And that's called bioprospecting. The other thing that's often done is to take a naturally occurring protein that does something kind of similar to what you want to do and make small changes to it. And so you get a protein that's very close to a naturally occurring protein, and you've hopefully modified it to do a little bit closer to what you want it to do. But what I'm going to talk about today is different. I'm going to talk about building proteins completely from scratch that carry out new functions. And the advantage of this is you can build the protein to do exactly what you want to do. You don't have to worry about what the closest that, that nature got, or try and modify something that's already very complicated. So I'm going to begin with a very brief history. The first stage in de novo protein design was understanding how to build new protein structures from scratch. And this really culminated in 2003 with the work of Brian Coleman designing a brand new protein which had a new structure and a new function. And since that time, a growing and now quite large community of people, really wonderful scientists, a number of whom, quite a few of whom are here, have been working to both design new protein structures, but also to design new protein functions, and in particular design proteins that solve current day problems. And in very recent years, this field has sped up with the advent, and you'll hear a lot more about this in Demis's talk, of deep learning methods. So I'm not going to tell you much more history in my talk. I'm going to basically sort of fast forward to the present. I'm going to tell you about the deep learning methods that we have been developing for protein design, and then I'll give you examples of what protein design can now do. So we've been developing generative methods for creating new proteins, and these methods, or the one I'm going to tell you about, called RF diffusion, is really inspired by methods for generating images. And these methods work by taking a large set of data, for example, images off the internet, adding different amounts of noise to the images, and then training a network to remove the noise. And once this has been done, if it's been done well, one can start with completely random pixelated sets of pixels, successively denoise, and generate an image that looks like it's an image off the internet, but in fact it's a completely new image. And in the same way, we took all the structures in the protein structure data bank, and I need to highlight that everything I'm telling you about from now on absolutely relied on the work of the tens of thousands of researchers who solved the protein structures in the PDB, because that's the data that we used to train. And also the people who organized the protein data bank, it was really an absolutely invaluable resource. So we took all the structures in the database, we added increasing amounts of noise, and then we trained a network to predict what the denoise structure at each stage was. So at the end, just as in the case of images, we could start with a random, completely random image and generate something that looked like a very plausible image. We could start with completely random configurations of amino acids, progressively remove the noise, and generate a new protein structure. And we found that these proteins folded up pretty well. We could make them very well. We could make them in the lab, but they didn't do anything. Now, in the case of images, as you all know, you can you can guide or you can condition the generation process with, for example, a text prompt. So you can say, generate an image of a cat sitting on the table, and that's what you get. And we can do exactly the same thing here, because it's not very interesting just to make new structures. What we want to do is make new functions. So we can condition this protein generation process on the function that we want to make. And one way to do that is shown here. This is the insulin receptor. We can carry out this generative diffusion process in the presence of a target, like the insulin receptor. And during training, the generative method has learned that, that proteins bind to other proteins through very shape complementary interfaces. And you can see that as this protein gets built up, how the shape match of this new protein matches the target. And we've now used this to design proteins to bind to well over 200 different protein targets. It's pretty much a solved problem now. I'm not going to tell you about all 200 of them, but I'm going to give you some examples of how one can use these binding proteins to solve problems in a wide range of different areas. And I'm going to tell you about 3 types of applications today: in medicine, in technology, and in sustainability. And I'm going to begin by talking about applications in medicine. So snake poisoning is still a major medical problem, particularly in developing countries, and snake toxins interfere with really fundamental biological processes. Now, an antidote for snake venom needs to be a very stable protein or set of proteins. It has to be cheap to manufacture, because where they're needed is in countries where there isn't necessarily a cold chain. You need them out in the field. You need to administer them almost immediately. And we thought this would be a good match for protein design, because the proteins that we design almost always are very, very stable proteins, so you can produce them in large amounts quite cheaply. And so here is a designed protein which is binding to the toxin, which is here. And all this work is highly collaborative. And so our collaborators in this case tested to see whether this designed protein could protect animals against the snake venom. And so in this experiment here, the animals who got the snake venom alone died almost immediately, whereas ones which got the designed protein were completely protected. And so we're very excited about this as a general way forward for generating anti-venoms that can be more effective and much cheaper than in current approaches. Now, a major problem in current medicine is inflammation, which is associated with many autoimmune and other disorders. And central to inflammation is a protein called the TNF receptor, and it's actually the target of many drugs currently on the market. And so here you see a diffusion trajectory conditioned on the structure of this target, and you can see how it's building a protein which is really exquisitely shape complementary to the target. And this protein is very effective at blocking inflammation in animals. So these are animals that have been exposed to an inflammatory treatment or inflammatory signal from bacteria. There's a very large amount of inflammation. Current drugs like Enbrel that are used to treat inflammation have some effect. This designed protein, because it binds so tightly, is even more effective. And so we're very, very excited now about making drugs to treat a wide range of autoimmune diseases and inflammatory diseases. Now, cancer, of course, remains a pressing problem, and we've been designing proteins to activate the immune system to treat cancers. And so this protein here, this designed protein, is bringing together two immune receptors, and this results in a very strong activation of the immune system. And again, with collaborators, Mike and Stephanie Dugan, they have been looking at pancreatic cancer, which is one of the most difficult forms of cancer to treat currently. And what they find is that this designed protein is quite effective, more effective than other known treatments in reducing the size of tumors in this model. And again, we're very excited now about getting this protein out in the world for into clinical development. Another important application of designed proteins is to make antivirals for pandemic preparedness. So this is the influenza virus surface protein, and here you see the diffusion process generating a binder here. But here we've added an additional conditioning criterion, which is we want this protein to be an antibody, which is a particular type of protein fold. And we wanted to do that because antibodies, the pharmaceutical industry knows how to develop antibodies, and so they like to work with them. And so here you see an antibody-like structure being generated that binds to the target. And here I'm showing you an experimentally determined structure of this designed protein bound to the flu virus protein. And you can see that in one of these colors, let's see, the experimentally determined structure is in purple, the design structure is in gray, and you can see they're almost identical. And that those of you who know about antibodies know that they recognize their targets through CDR loops. And these loops, which are being built completely from scratch, in addition to the docking mode, are coming about almost exactly in the design model. So I think that antibodies are currently discovered using by immunizing animals or pulling them out of people. I think we're now seeing a transition over to using these kind of design methods to generate antibodies. So another sort of increasingly important medical problem are neurodegenerative diseases like Alzheimer's disease, which are associated with the formation of long amyloid fibrils. And there are several proteins, among them amyloid beta or A beta and tau, which form these fibrils that are observed in the brains of people with neurodegenerative disease. So here what we've been doing is designing proteins that will bind to the disordered portions of these proteins that would normally interact with themselves to form amyloid. And these trap these regions so they can't form this extended amyloid. And this experiment, again with collaborators, is looking at the, if you simply take these proteins, put them into solution, they aggregate as the amyloid is formed. And with these trap proteins, this amyloid formation is completely blocked. So this is measuring the extent of amyloid or aggregation that's going on. And so we're very excited about these as a possible way forward with neurodegenerative disease. So there are many, many other applications we're excited about now in medicine, but I'm going to switch over and now talk about technology. So one general problem in technology is in sensing. How do we sense arbitrary molecules, and how do we determine sequences of things like DNA and proteins? And in nature, there are channel-like proteins or pore-forming proteins that look something like this, and people have adapted those for problems like sensing and sequencing. But they aren't really optimal for that because they didn't evolve for it. And so with Anastasia Vorobyeva's group, we've been developing methods for designing new pores completely from scratch, and this has the advantage that we can completely customize all properties of them, of the pore and so forth, to be optimal for the task we want to carry out. And this just shows you some examples here. These are 3 designs with increasing pore sizes, and the larger the pore, the more ions that can flow through it, and so the larger the current that is measured going through these. Now, to turn these into sensors, and we're very interested in constructing molecular noses, what we do is to design proteins that bind small molecules that we want to detect, and we can put these proteins on top of the pore, like shown here. And then when we put this combined protein into a membrane and we measure the current across it, we see a sizable current. When the small molecule is added, basically it closes up the channel, and that's shown here, and then the current completely shuts off. And this can be measured because one can measure electrical signals very, very sensitively. So we're excited now about being able to make very general molecular or electronic noses that can tell you what's in a solution by integrating the information from many different pores like this. Now, what you'd really like to do is to couple this kind of sensing scheme with electronics directly. What I showed you before was those were inserting into a hydrophobic lipid layer. So we've been working on methods for inserting these sorts of pores into silicon nitride chips, so they could ultimately be in, for example, in your cell phone to tell you what's around in the environment. And so this requires a totally new class of pore, which is an example of which shows here. We want things, of course, proteins never evolved to interact with silicon nitride or to insert into it, so we have to design new types of protein structures. And here are some examples of EM structures of sort of this gallery of pores that we've made to try and to approach this problem. And again, with collaborators, we have, this is from Jens Gundlach's lab at the UW. So here we have a pore in a silicon nitride chip. There's a lot of current going through. The design protein pore can insert in, and we get a very stable current. And now we can start modifying these pores, as I showed you with in a moment ago, by incorporating binding domains and other sorts of domains on the interior to be able to read out sequences and other properties. So another general problem in technology is targeting very specific sequences of DNA. So, you know, our genomes have lots and lots of DNA in it, and there are many applications, for example, fixing genetic diseases where you want to target just a very, very specific region of the DNA. And so to do this, we've been designing proteins which bind to DNA and have very high sequence specificity. So the protein shown here makes very specific interactions with the DNA bases that make it very sequence dependent. And this is now some experimental data showing that if you take this protein and measure its binding to DNA, it's very sensitive to any changes of the DNA sequence. So when the sequence is the red color means that the binding is weaker. So these sorts of designs can read out DNA sequence very precisely. And we're excited about using these, for example, to create new ways of smaller, more compact ways of fixing genetic lesions and other activities. So now we have a very exciting, Neil King had this very exciting result last year with the first approved de novo design medicine. And this is sort of an interesting case where Neil started off making these self-assembling nanoparticles sort of just to see if we could do it. And then after he had succeeded in building them, he realized that he could make vaccines out of them by putting pieces of viral proteins on them. So during the pandemic, he put a part of the receptor binding domain of the COVID surface protein on these, and found that this elicited really, really strong immune responses. And this is now a clinically approved medicine. It's in use it's been approved in several countries. Here is the designed protein in a vial, and we're very excited about, a year or two from now being able to have many, many examples like this. Now, these sorts of nanoparticles are very useful also for delivery, but there's a problem in that if you want to deliver a lot of material, like say a drug to a specific site in the body, the size of the container matters, and with a bigger container you can make more. So this is an interesting geometry problem. The icosahedron or dodecahedron is the largest completely regular polyhedron. So if you want to, you can make it bigger. So this is 12 pentagons coming together. You can make it bigger by inserting hexagons between the pentagons, as shown here. This is just as in a soccer ball. But here you have a problem because you can see the angle inside the pentagons is different than the angle inside the hexagons. So we've been trying to figure out how to solve this problem, and there are basically two solutions I'm going to show you here. These are both electron microscopy structures of designs. In this case, we've solved the problem by using just one type of protein that can adopt different conformations in the different sites in this nanoparticle, allowing the interspersal of pentagons and hexagons, as you can see here. In this case, we've used different protein chains that sit at the different non-geometrically equivalent positions. And the advantage of this is that both of these methods, and again, this is experimental data, cryo-EM structures, give much larger particles than one gets. This would get with pure symmetry. This, for example, is one of the most widely used current delivery vehicles based on a naturally occurring virus, AAV. Now, everything I've told you about so far is kind of static, but in technology, we'd like to be able to turn things on and turn things off. And so to do that, we've designed proteins that undergo conformational changes and have two different states. So on their own, they fold up to one state, but then when an effector is added, they convert into another state, as shown here. And if we build now larger assemblies out of these, like this triangular one here, in the presence of the effector, these will now bend, and so we'll get, and now we can effectively convert, you can see when this becomes a right angle, we effectively convert a triangle into a square. And this works really nicely. We chose to go for these simple geometric transitions because it's very easy to follow by electron microscopy. So here's a design just like this one that was designed to be a triangle. Then the effector's added, and you see you get a square. And this is electron microscopy data. You can clearly see that transition. And here's another case where you have a square that's going to a pentagon when the effector is added. And for the biochemists in the audience, this is clearly allosteric control because this effector is not acting at the interface between these chains. It's acting quite far away and working through by changing the conformation. Well, that wasn't very particularly useful. It was sort of like that showed that we could build switches. But where might you want actually a switch? And coming back to immunotherapy, we might want to activate the immune system to treat cancer, but then we might want to turn it off because overactivated immune system can be a problem. So here I'm going to show you an off switch. Here's a design protein that brings together two receptors activating the immune system, as I showed you earlier. But now we've built in a binding site for an effector which turns the whole system off. And here's a cartoon of how it works. So here's the design, bringing the two chains together. This effector comes in. It causes an allosteric change, which kicks off this other receptor subunit. And this is, again, a wonderful collaboration. This is actually going on the surface of a mammalian cell. You're seeing these two receptors are labeled with fluorescent dyes, and when we add the drug initially, the two receptors come together, and then when the effector's added, they come apart, as you saw here. And so we can see the receptors coming apart when the effector is added. And this has a profound effect on cell signaling. So when we add the drug, this protein, it brings these two together. We get an activation of signaling. And then when the effector is added, we can shut it off very, very rapidly. And this is useful not only for shutting off the drug when you no longer need it, but for probing exactly what the effect of having these receptors together for different lengths of time is, which has been kind of an outstanding question in immunology. And the final area I'll tell you about is sustainability. And some of the most remarkable proteins in nature are enzymes, and these carry out chemical reactions. And the way they do this is they have these very large, complex structures, and if you zoom in, they have active sites where there are a few key amino acids that are mediating the chemistry and carrying it out on a bound small molecule. And so we can now design catalysts completely from scratch. We do that in kind of the opposite way. We start by specifying the active site geometry, what the side chains on, where they should be, and the small molecule or transition state we want to act upon. And then we use the diffusion process to build up a protein around that active site. And so this is kind of neat because we can explore in nature, and biochemists and enzymologists have been doing this for many, many years. You just have one thing in your enzyme, and you can sort of tell stories about how it works. But with this buildup process, we can change what the active site, what the side chains are in the active site, and what their geometries are, and look at the effect on catalysis. And so we can build up sites of increasing complexity, and what we find is as we adjust the geometry and make the sites more complicated, we get increased activity with a sort of with a partial site. We get enzymes that carry out a single round of the reaction but then stop. If we build up full active sites with sort of more extensive networks of interactions, we get quite active enzymes. And we can solve the structures of these by X-ray crystallography, and then we can see to check to see how accurate our designs are, and they can be extraordinarily accurate. This is now a superposition of the experimentally determined crystal structure and the actual design. And the activities of these are much higher than had been found previously straight out of the computer. That was for a complex multi-step reaction. We can also use metal ions to carry out catalysis. Here, this is actually starting with a quantum chemistry calculation of the reaction pathway. And we identify the transition state on this pathway. And then we basically build a protein around the metal and the binding site. And this is sort of a blow-up of the site. And as I said, in all cases I've shown you, we are making synthetic genes, encoding the designs, putting them into bacteria and testing them. Here we tested 96 designs, and we got a number of active designs, the most active of which had very, very much higher activity than anything that had been made straight out of the computer before. And this is starting to approach the activities of native enzymes for this type of reaction, which have evolved for hundreds of millions of years. So there are many problems. This is a bond-breaking reaction, and there are many problems where this is important, including plastic degradation. So we're now building sites, completely synthetic enzymes, to break down plastic and other polymers using exactly this type of active site. Now, one of the other remarkable things that proteins do is to harvest solar energy, and this is done, of course, in the photosynthetic process, which is very complex, which involves very big proteins, but they have at their core what's called the reaction center, which is made up of 2 chlorophyll molecules. So we've been, again, kind of building from the outside, from the inside out. So here we've designed a small protein that binds 2 chlorophyll molecules, exactly as in the reaction center. And now we can start building this into larger assemblies. And so this shows taking this protein here and now designing it into these cubic assemblies. Each one of these has 2 chlorophyll molecules. And so we can start building up completely artificial synthetic photosynthetic systems. And we can also now tune the absorbance profile to cover wavelengths which are not well covered by natural photosynthesis. And my final example, again, something that nature never tried to do, is in mineralization. So in nature, we have things like bone and tooth and shells where proteins are directing the mineralization of calcium carbonate, calcium phosphate to make these really remarkable hybrid materials. So we've been really fascinated by this, and so we've been approaching it by designing proteins which have arrays of amino acids that we predict would drive the nucleation and the mineralization of inorganic compounds. In this example, though, we chose not to apply this to calcium carbonate or calcium phosphate, but to zinc oxide, which is a semiconductor. Because you can imagine if you could template semiconductor growth with proteins, that would open up a whole new range of interesting materials. So here we've lined this a part of this structure with this interface, and when we incubate this with zinc oxide, we get zinc oxide filling this part of the triangle, as you can see here, which is opening up really very exciting new ways of making completely new hybrid materials. So I've kind of given you a bit of a whirlwind tour of some of the things that can be done with modern protein design. The applications, there are really a wide range of applications in medicine. I've talked about pandemic preparedness and cancer immunotherapy, neurodegeneration. There's applications in technologies I've showed you, and many interesting problems of sustainability. And my prediction will be that over the next 5 to 10 years, we'll see an increasing number of designed proteins out in the world addressing these problems. So finally, most important part, I want to thank all the amazing people who contributed to this work. It's really, I mean, the prize really celebrates the work of a really large community of people currently in my research group, and then a very, very large community of former students, postdocs, colleagues, and friends. And it's really been just absolutely wonderful to work with everyone. And this prize is really all for everyone who contributed to this. And I'm really happy because I think nearly 200 people have come to Stockholm to help me celebrate this, which is really, really wonderful. And I hope I get some time to actually see them. So thanks.